Okay, hi everyone. Welcome back to our afternoon session. So we are once again very happy and privileged to have Professor Herbert Levine with us. So he's been introduced before. So very briefly, Herbie did his undergrad at MIT in physics, and also PhD in physics at Princeton, and then he moved on to do a postdoc at Harvard. All of this was in physics, right?
Everything in physics.
Everything in physics, and much later in life, he switched on to doing biological problems. So all of you physicists...
Again, from the perspective of physics.
So all of you physicists here who keep asking, can we switch to biology? This is the best example and motivation that you can see. Thanks a lot, Herbie.
Okay, so I decided to change what I was going to talk about. Originally, I was going to give a sort of introduction to networks and EMT, and that's the last thing in the world you need to hear about again, because I think you've already heard enough about that. So I decided to talk, and this will be the first of two talks, I'm giving a second one tomorrow morning, on an aspect of the problems that are related to cancer, but more generally related to how all of biology works. In particular, I talk about transcription, but the philosophy is more general, that I think has not yet been sort of presented at this meeting. And the philosophy is the following. We've heard many, many talks about networks, about states, about transitions between states. The talk this morning was a very good example. It started out by being about shapes of cells, and then all of a sudden, instead of really being about shapes of cells, it was about sort of how you take this data about shapes of cells and analyze it using very interesting methodologies from a sort of data science perspective. But nowhere in that talk did you ever hear about how an EGF signal coming into the cell reorganizes the cytoskeleton of the cell, provides forces on the cell membrane to actually change the shape of a cell. So there's a whole layer of biology which is not just information. It's the part where, biology has to exist in the real world. It has to do things to cells. It has to give them shape. It has to allow them to move. It has to allow them to respond to chemical signals. And that part is equally valid part of what you need to be able to understand to work on a biological problem, as is the information theoretic side, at least in my opinion. So I sometimes call this my philosophy of genes are not genies. So, a genie is something that magically says something, and, a genie says, make an arm, and all of a sudden the embryo makes an arm. Well, it doesn't quite work that way. Going between what the gene does and breaking the proteins, instantiating a physical chemical reaction that then gives rise to the actual structure and makes that structure functional is the other side of the coin. So I'm going to talk about that aspect of at least the problem in the concept of transcription. And so I'm going to talk, really the talk has two parts, which you'll see. The first part really is an attempt to show you that sometimes physics can actually be necessary to understand something about a biological system. And the second part will be returning a little bit more to chemistry, to the issue of chromatin marks, which then is absolutely clear has a critical role to play in cancer and every other biological thing that we've been discussing. Okay, so that's the outline of the talk. So the first part is about, well, here is the structural schematic of what DNA looks like. And I've just chosen a eukaryotic cell for convenience. And so DNA is not an abstract thing that is an information storage device that you read and that mutates and that you read out and make messenger RNA, et cetera, et cetera. It's a physical thing. And sometimes the physical thing has an effect on what you care about. So here's a picture of DNA. I mean, you've all seen pictures like this. This is the double helix. Then on a larger scale, it's wrapped around these things called histones. These are proteins that make little balls that are sort of like spools where the thread is wrapped around the spool. The DNA wraps around each histone about 2 times or so. And then these, these ball-like things, this is called a nucleosome, are then, sort of packaged in some nice structure. Maybe it's this, here it's shown to be very ordered. It's not clear it's really that ordered. And eventually you go into the scale of the chromosome, which is, obviously what you see when you're looking at things at a very, very coarse magnification. Now, because of the physical nature of this whole thing. This is, this is the major player when you're talking about how you're going to be making RNA and thereby making proteins and thereby determining what cells are doing. The physical nature of this object very often, and the chemical nature, obviously can play an important role in what's going on. And so I'm going to give you sort of two examples of this. The first, I'm going to talk about something called supercoiling and its role in transcription. And this is really the physics part of what I wanted to say, because I feel that it's good for you to hear about at least some physics. And this is, if you want to read, there's sort of two papers on this. One that we published about 1.5 years ago, and one that's submitted, waiting for Sumitaba to finish looking for a job, maybe getting one, hopefully, and to go back to finish the last calculation to respond to the referees. So this should appear sometime this year. And this is about supercoiling and transcription. And then I'll talk about the chemical part. So the supercoiling is about the physical structure in space of this loop that you see here. And then we'll talk about histone marks. Histone marks are post-translational modifications of these protein constituents of these spool-like objects. And those protein constituents are called histones. 8 of them make up the nucleosome. Each of those histones has a sort of tail, sort of wagging out away from this thing. And what's well understood, although not in any quantitative way, is that post-translational modifications that go on this tail, adding methyl groups, adding acetyl groups, other things you can do, can have a very large effect on transcription, because they control how easy is it to, first of all, reach the DNA that you need to attach to begin transcription, and also how easy is it for the polymerase that makes RNA out of the DNA, copies the DNA into the RNA, how easy is it for it to progress down? It's got to make its way through this sort of jungle of obstacles, and how easy that is depends on these histone marks, sometimes called the epigenetic code of the cell. So I want to talk about those two different pieces. We'll start with the first, and then this is a part which, from the point of view of cancer, is a lot more important, but is also a lot more complicated. And also, we actually know very little about it. So let's start with supercoiling. I have here my example. Here is a loop. And what happens when you try twisting it? So you twist it, and you twist it a little bit, and nothing happens. And then you twist it more, and all of a sudden, this thing stops being just twisted. It starts creating a sort of 3-dimensional structure. It sort of leaves this 1-dimensional world and sort of bulges out into the 3rd dimension. These bulges are called plectinemes. They are loops where the coil goes around itself. Here you see a nice plectineme over there. And these things are physical structures that can occur on the DNA. So the first thing you can do, of course, many people just interested in protein biochemistry studied these effects. So you can look at just this basic effect, and the way to think about it is, now DNA is a little bit different than my rope there, because my rope there starts out with no twist whatsoever, and then you give it a twist and it starts recoiling. If you would try this experiment by yourself, you're welcome to borrow my physical example. If you try, you'll see that it resists being twisted. So it's like a spring. You try to stretch the spring, and it resists being stretched. If you try to twist one of these things, it resists being twisted. So the technical way to describe that is that when you have to apply a torque, a twisting equivalent of force, in order to make it twist. You have to impose energy, you have to add energy to the system, because twisting it's not its natural state. Well, DNA already has a natural state, which is a little bit non-trivial, because the DNA double helix actually starts out where the 2 different strands go around each other. That's why it's a double helix. Each strand wraps around the other strand with some typical typical spatial distance of how long it takes to go around one turn. So for DNA, typically one total turn coming back to the same is about 10.5 base pairs for normal DNA. So twisting DNA means, if you do the same thing, if you grab onto DNA and you twist it, which you can do in experiments in single molecule biophysics labs, means you're changing that number. You're putting in more twists, so now the DNA twists faster and it goes around in less than 10.5 base pairs, or you can undertwist, you can go the other way, you can put it the other way, and in that case, you get the opposite effect, and either of those costs you energy. Both of those are resisted by the cell, by the DNA. The DNA wants to be twisted at a certain rate based on its chemistry and base pairing and all that stuff that was worked out a long time ago. And so either twisting it this way or that way, you get a restoring torque that you have to work against in order to keep twisting it. Okay, and you can actually derive some very simple formulas. Here are just some pictures, and this is a picture of what some of these different supercoiled states look like. These are plectonymes, and in the case of DNA, where you see these, that you have the double helix, then you also have this sort of super helix, wrapping the whole thing around itself. And in general, what happens is, if you do a little bit of twist, all you do is add twist. If you do a lot of twist, then you start making these very complicated nonlinear structures. And the two ones that you can make is, if you over twist DNA, what you do is you create these plectonymes. If you undertwist DNA, then what happens is you create melted pieces of DNA where you've undertwisted it and the two strands actually separate from each other over some distance. So here is an example of melted DNA where you've twisted this, and now in order to relieve that energy, in order to come to a better state from the point of view of the energy, it actually has melted the DNA in this region over here. Okay, now one more thing about biophysics, which, yes? Yeah. Yeah, so I will get to that. But, so first of all, sometimes DNA does exist in a loop, for example, in plasmids and things like that, or circular chromosomes. In long mammalian chromosomes, it won't, but there's things that are equivalent to that. There are places that are attached to very heavy things where the DNA is not allowed to twist. So it's like having, it's not a loop, it's not periodic boundary conditions, but under many cases you can get situations where there's zero twist boundary conditions over some distance of a few kb or something. So yeah, I'll say that again in a moment. Okay, so you can derive, and again, we're still in the realm of doing biophysics and in single molecule labs, and not talking about cells and not talking about transcription at all. We're still at the level of saying, gee, this is a fun polymer, let me play with it in my lab. You can attach beads to it, you can twist one end versus the other, you can make all these measurements. And this is a theory that was done a long time ago of what happens when you do this twisting. So you measure twisting by essentially what's called the supercoiling density, how much you've sort of twisted it per distance. And you get a response, and the response is a little bit complicated. The response that you expect, if you learn freshman physics and you learned about springs, you learned that in a spring, you get a linear response. You pull the spring apart by some distance, you increase its length by some distance L, and it resists that. It provides a force acting against you, which is proportional to the spring constant times L. And if you want to twist it further, you have to pull against that force. So the twisting, or the pulling of a spring gives rise to a linear response. Now, there's a region here where it's the same. So until you get to this sort of interesting nonlinear, plectoneme formation where all sorts of new things start happening, then, you get a linear regime. So here is the linear regime over here. This region between, down here, 0 down to, some over this region over here, it's linear. The torque, which is the equivalent of force when you're talking about rotating something, is over here. It's, just a linear function of the supercoiling density. It's the equivalent of a spring constant, force being proportional to extra length. Yes? Well, this is universal. I mean, this is, basically it's universal. This is true because the DNA is a polymer at the end of the day. It's not because it has a code for anything, right? So it's not related to any of the biological function of DNA. It's just related to DNA as a polymer. So if I take a polymer and twist it, I'm going to get something like this. Now, the part that's a little bit related to DNA that's not related to a normal polymer is the fact that it has this double helix structure. So the double helix structure shows up down here, which is, I told you, if you have this negative supercoiling, if you untwist the DNA compared to its normal helix, it tends to melt. So that's this part of the curve here. This is twisted DNA interacts with melted DNA, and this part is the plectinine part over here. So anyway, this is just a result which has been measured in the lab, and you can go out and discuss it, and it's great. And the question now you're all asking is, what does this have to do with transcription? Or what does this have to do with genetic circuits or cell fate transitions or anything like that? So that gets us to the next slide. Whoops, it would get us to the next slide if I could remember to push the right button. So let me try to create a picture for you. Imagine that you're trying to go along, and I don't know if they have these in India, but in the US they have these roller coasters. You know what a roller coaster is? Okay, good. I have no idea how these things cross cultures. And some of these roller coasters have twists in them. So, instead of just going up and down, you have ones where the car, in order to keep going, is going along a path that looks like a helix. Now, in order to go along a helix, you're a person. If you want to go along a helix, then you have to rotate, right? Because you're starting like this, and you're going this way, and you're going that way. So you have to rotate. Now, the mechanics of biological systems are a little bit different than what we teach our freshman physics students. And our freshman physics students, we teach them Newtonian mechanics, right? Newtonian mechanics, F equal ma, forces are proportional to acceleration. You can keep going at constant velocity forever if you don't apply any force. Unfortunately, no biological system works in that regime. All biological systems work in liquid media, room temperature, and they all work in a regime which we tend to think of as viscous dominated or dissipation dominated. It's like throwing a rock from a high building, not through the air, but through jello or something like that, or through some very viscous material. The law of physics is not F equal ma. The law of physics is F equal mv. It's not called m, but it's the usual law of biology that's relevant is that forces are proportional to velocities. Left on their own devices, if you have something which is moving, even a cell, and you stop pushing it, the cell will stop. It will not behave like Newton's equation tells you it will behave, which is it will keep going. It will behave because it will be moving in some type of fluid. It will pick up friction from moving through the fluid, and as that friction builds up, it will stop the motion, suck energy out of the motion, and cause the object to stop. If you want something to keep moving, you have to keep pushing it in the real world of biology. That's for cells, that's for molecules, doesn't matter. All biology works in that regime. Inertia is almost always irrelevant. Now, if you think about applying that to this, going around this roller coaster, you're going around the roller coaster, you're rotating at a constant rate. So if you were doing this, in the vacuum in the freshman physics world, you would be able to keep rotating at a constant rate without applying any force. But that's not the world in which biological molecules live in. The world in which biological molecules live in is a world in which you need to apply forces to keep going at constant speeds. So you have to apply some type of torque. You have to be able to push to keep going around in a circle. Now, a roller coaster is made out of steel. So however hard it is for you to be pushed going around in a circle, it's much, much harder for you to actually twist the roller coaster, right? I mean, the roller coaster is not going to twist because you're pushing on it. So even though, when the roller coaster is applying a force on you, trying to make you rotate, and you're applying the same force in reverse on the roller coaster, trying to make it rotate, the roller coaster is not going to twist. But DNA and RNA are much more closely related to each other mechanically than you are to that roller coaster. So the answer is, whenever you transcribe a piece of DNA, the RNA polymerase, which it comes, attaches to the DNA, has to move along the DNA. The RNA polymerase has to do this in order to keep moving around the DNA, because the DNA is a helix. And if it has to do this, it means something has to be providing a torque. And just because Newton's law, third law, says that every action has an equal and opposite reaction, if you push on one thing, it's going to push back on you. When you're pushing on the RNA, then by definition, the RNA is pushing on the DNA. But the DNA is able to twist. it's not going to twist, it's going to require a lot of torque to twist, but it's going to be able to twist. So the real equation when you're doing transcription is there's a balance between how easy is it for the RNA to go around in a circle versus the RNA going straight and twisting the DNA ahead of it so that it doesn't have to go around in that circle, right? If you imagine the RNA just having this, be able to twist the DNA in front of it such that it's always going in a straight line, then you wouldn't have to pay that price of forcing the RNA to move in this complicated path. Now, the reason why this is important is, RNA polymerase is pretty small compared to DNA. And so, it may be most of the time, if you have really just RNA polymerase, it would be easier for the RNA to rotate. The problem is, well, it's not a problem, but the reality is RNA polymerase is making RNA. So out the back of the RNA polymerase, attached to it, is this nascent messenger RNA molecule, which can be pretty big, and you have to turn that too. So as the RNA is progressing, you actually get more and more resistance to going around, and therefore more and more ability for the RNA to be twisting the DNA rather than vice versa. So you can take this conceptual idea and you can make a simple physics-based model of how that works. And here is the physics-based model. It says the following, and I don't want to spend an infinite amount of time going through all the detail. Whoops. Yes, please ask. Yes. So you're always one slide ahead of me. You're always one slide ahead of me. So that will be on the next slide. So the answer is yes, but then it depends on how much top-line isomerase you have. That's exactly, okay. So fine. So the equation looks something like this. I'm not going to, worry about the details, but the equation basically says you have two things that can happen. You can have the RNA. This is the RNA with the sort of messenger RNA coming out the back of it, which can be turning around, or you can have the DNA, which is being twisted, and these things are related to each other and related to the amount of torque you have in the system. And I already told you that you had a piece of the torque. You could calculate the torque if you knew these things. Now, just to be very clear about the previous question that Shaon asked, so the idea is if you have twist, the DNA, if you have a twist, it means that you have a piece of DNA, let's draw it as a helix, and over here you have phi is some non-zero number. You've twisted it by some angle. Now, if the DNA were not attached to anything, then you can twist anything, if it's not attached to anything. I can twist this piece of chalk forever, if nothing happens, who cares? If, on the other hand, somehow I epoxy the piece of chalk to my fingers over here, and then I try to twist this, then, of course, it can't twist at the ends. It's got to twist in the middle, which means I create a profile which looks like this, that I have a high phi over here, I have low on the two ends over here. On this end, I have positive supercoiling, which happens to be defined in sort of minus d phi dx. And here, I have negative supercoiling, which happens to be plus d phi dx. So this, in the biology world, was understood in a qualitative sense, I think. I don't know if we were the first people to write down an equation for it, but it was understood. It was called the twin domain model. So there were two domains that were created by this process of having the DNA pick up some non-zero angle in the middle, but making a gradient of this angle, so therefore it costs you energy. And the twin domain model was in front of the progressing RNA polymerase, you'd have positive supercoiling. Behind the progressing polymerase, you would have negative supercoiling. Now, Shaon's question was, where do these boundary conditions come from? And I think it's not completely known, but we'll see actually as we go on later that if you have very bulky proteins that are attached to the DNA, then they can, they're very hard to twist, and they can effectively impose zero twist conditions. And we saw that in some particular example, which we'll talk about. That will be the end of, but that's sort of the idea. And if you have a periodic system, then it's fine in a sense. But if you have, or you can impose it by hand in the lab if you make DNA that you can attach to beads and prevent it from rotating at the two ends. But in the real world, there's some phenomena having to do with looping and nucleosomes or whatever that can prevent over some distance DNA from just uniformly rotating. Okay. Now, where am I? Now, so what is the total equation? What is the total picture now that governs what's going on here? So there are two other things I need to tell you. One is I told you, which is already, I sort of told you already. The first, I have an equation that tells me how to compare how much the RNA is going to rotate versus how much the DNA is going to rotate. That's this equation here. I have a constraint that says if my polymerase moves a distance x and I have a twist in the DNA, then that's got to either be gotten by rotating the RNA or by rotating the DNA. There's sort of a constraint on moving is related to angular motion because I'm moving along this helical structure. That's this first equation. And then I need to know something else that was measured in the laboratory of Michor, which was that if I have polymerase that's moving into a region that is supercoiled, that has a high torque, then it's harder for the polymerase to actually proceed. That the polymerase makes it, it's more difficult for it to move if it's moving into DNA, which has a significant twist. And this is a phenomenological fit to, their equation. It basically says that the DNA polymerase would have a normal velocity of v0, and then depending on the torques in the system, if you went to the limit where the torque in front was bigger than the torque in the back, then eventually the velocity would go to 0. So RNA polymerases are inhibited from moving by having torque in the system. Now, there's one other piece of the story that I have to say, and this was Shaon's second question. The second question is, if I just let this go on its own, did this in a laboratory and just put this material and let it go, well, what happened is the RNA polymerase would eventually stop. It would become more and more bulky. Eventually, the torque in front would build up because I'd be more and more twisting the DNA in front. And so I'd build up more and more twist, more and more things, basically opposing the motion. And eventually, the RNA polymerase would just stop. So why doesn't that happen in the cell? Why doesn't the RNA polymerase just stop after a while? And the answer is because there is an enzyme which is called topoisomerase. Topoisomerase is a well-known enzyme in the cancer community because there are many, many attempts at using topoisomerase inhibitors to fight against cancer. I don't think they work very well as far as I know, but nonetheless, it's been explored in great detail. A topoisomerase is a very nice name for an enzyme that changes the topology. And in some way, which depends on the magic of chemistry and molecular biology or whatever, the enzyme is able to take pieces of DNA that have this wrapping around each other and essentially pass one loop through the other. They're able to remove twist from DNA. And it does that in some very complicated way. It's got to break one part of the DNA and pass it through and then re-anneal it and get rid of the twist. So if you dump a lot of topoisomerase on your system, the system doesn't care about this problem. The RNA is going along, it's twisting the DNA, the topoisomerase is coming in and untwisting the DNA, and everybody is happy. But of course, there's a rate consideration. If you have something which is moving quickly, then depends on what the rate constant is of the enzymatic activity. If the enzymatic activity is too slow, then you won't be able to keep up with this motion, and you might see the effect of the RNA polymerase slowing down. And if you have enough topoisomerase, now what is the rate? I mean, rates as a number, it's got to be slow or fast compared to something. And if you think about it, what it's got to be fast and slow compared to is how often you're actually coming along and beginning to transcribe the gene. So if you have a promoter which is very active, very highly expressed gene, very active promoter, you have lots of RNA polymerases going by one after the other, creating this positive supercoiling, then you probably won't be able to keep up. If you have a very slow acting promoter with a low transcription rate, then you probably will be able to keep up. So that's the logic to think about, that this is an effect that's going to be more important for rapidly transcribing genes rather than non-rapidly. Okay. Now, okay, so here is the picture. You have this RNA polymerases that are moving along, and they're leaving behind this trail of nascent RNA, and it's twisting the DNA. Now, yes? Yeah. Oh, yeah, I mean, whatever at the end is being, yeah, so you need something at the end which is resisting being twisted by a lot. It's included, in other words, we don't include that in detail. We assume, and I'll talk about exactly one case where we think we know what causes this in a few moments, but we assume that there is something out there with some typical distance, and we actually then use that to try to figure out what the data, we use that to fit some of the data, which I'll talk about in a second. But there has to be something out there, because if the DNA was just able to twist freely, then who would care? You wouldn't care. It would just twist, there wouldn't be any penalty, and everything would be fine, but it can't do that, even in linear chromosomes. Okay. That's one of the uncertainties of the model, by the way, is exactly how to think about those barriers to supercoiling. You know. Yeah, you can, in this model, you can calculate all those things. And you can calculate whether what type of thing would be able to stop, would be able to sort of provide boundary conditions. And then we have an example where we think we understand that that's actually what happens. And I'll get to that in about a little bit of time. Okay, now, it's sort of funny because we introduced this model to explain a different phenomena that I'll get to in a moment. And it's a phenomena which is very relevant to a lot of the discussion we have been having about cell-fate transitions. But there was a consequence of the model and a consequence of this idea that we hadn't fully appreciated. I mean, it was there. We could have, we sort of, it's in the model, but somehow an experimentalist had to sort of point that out experimentally that this thing actually predicts something else. So I'm going to tell it to you as if we predicted this and then it was seen experimentally. In fact, we proposed this model. We used it to explain something else in a moment. And then some experimental group came along and did an experiment that we then said, yeah, the model should immediately explain why that's true. And then we showed that. So, okay, so it's sort of interesting that you don't always realize what your own theory is predicting. So what's the simplest phenomena that this theory or this idea predicts? So here is now a train of RNA polymerases. A train means that you have in many systems, the RNA polymerases come along, and you shouldn't think of it as one RNA polymerase comes along and finishes transcribing the entire gene, and then the next one comes along and transcribes the entire gene. There's no, at least for rapidly expressing genes, it's not the way it works. It takes some physical amount of time for the polymerase to actually make it across the gene. And in the meantime, other ones can start. So the typical situation for again, highly expressed gene is that you have a train of RNA polymerases, sort of one after the other, going along and translating. And transcribing the gene. No problem with that. Now, if you look at the picture that we have over here, then this RNA polymerase over here is leaving in its wake a negatively supercoiled DNA. This one is leaving in front of it a positively supercoiled DNA. The positive and the negative cancel each other out. So in between each of these polymerases, you actually don't have to twist the DNA. It actually cancels out. You're twisting it one way for the one in front, and you're twisting it back the other way for the one in the back, and the net effect is to not have a twist. So if you have a train of polymerases coming along, the front one up here, this one up here, is fighting against the headwind of going into this region where it's increasing the back reaction of the DNA. But the ones behind it get a free ride. It's sort of like all those pictures of birds in flocks, where the front bird is doing all the work, flying through the air, and then the next one is creating a sort of barrier where the next ones don't have to work that hard, and they're just sort of gliding along. And of course, what birds do with that idea is they just keep sort of cycling out, which bird is in front. Well, here, there's no way to cycle out which one's in front. So the first one is slowing down, and the others are not slowing down. Now, what that means is that you have two possibilities for what can happen. The first possibility is that depending on how many you have there, the average rate of transcription actually goes up compared to what would happen if you had only one. Because let's say you have 10 of these going at the same time. 9 of them are moving quickly, 1 is moving slowly. On average, you'll get through your 10 transcribed RNAs faster than you would if you didn't have this cooperative effect. This is a cooperative effect between separate RNA polymerases occurring through the mechanics of the DNA. The next thing that can happen is if you take that too far and they catch up to each other before you finish the process, you can get a traffic jam, right? So you can have the first one almost stopped, the next one is going along and sort of gets very close to it. Eventually, it physically overlaps with it and has to stop, and then you get a traffic jam that builds up. So the traffic jam builds up is what we originally were interested in because, and I'll get back to it in a moment, because the traffic jam buildup gives you some mechanism to increase transcriptional noise. What it does is it converts the transcription process into a bursty process. It says that in that limit where you have a rapidly transcribing gene, the noise is not just Poissonian noise, as you expect for most simple chemical reactions. Something comes along and binds and then unbinds, which you always get Poisson distributions and Poissonian noise. It actually gives you bursty transcription. You get bursts of transcriptions, then the material runs into a traffic jam and stops. It waits for the topoisomerase to come along. Waits meaning on average, it has to wait some amount of time before enough topoisomerase action comes along to relieve the traffic jam. The traffic jam clears, you start transcribing again, and then after a while you go into another suppressed phase. So that's an exact model for what's called bursty transcription in the RNA world. And in fact, it's well known that if you look at data, transcription for various genes really is bursty. It's not described by a simple process of RNA binding to the DNA at some rate and then just moving along and transcribing and then RNA decaying at some rate. If you solve that problem, and maybe I'll write down the equations just to show you that we also can do Markov chains like everybody else, then you get a Poisson distribution of noise. Poisson distributions are the baseline of what you expect noise to be. If you consider bursting, bursting is noisier than Poisson distribution. Bursting is what happens when you're waiting for a bus on the city streets. You've been waiting for 20 minutes, and all of a sudden 3 buses show up at the same time because there's a process whereby those buses actually attract each other. How do buses attract each other? Well, think about it. The first bus is going along, and there hasn't been a bus for 20 minutes. So there's a ton of passengers that keep getting on. There's a lot of headwind for that bus. Every stop takes a long time. The other buses that are right behind it don't have as much headwind. There's not as many passengers. So they go through the stops more quickly. They catch up to the first bus. Pretty soon you have a whole posse of buses going along, and your arrivals of buses are bursty. And instead of waiting 10 minutes for each bus, you wait 20 minutes and then 3 buses come. So that's what RNA is doing in this model. But you also have this other effect that when you don't reach that pure traffic jam limit, you actually get this cooperative effect. So here is an experiment done. This was done in bacteria, essentially showing this effect. So this is the title of their paper: long distance cooperative antagonistic. Long distance means several kilobases here, just so you know what long distances means in the molecular world, right? This is not microns or millimeters or something. It means, but but it's not one base either. It's not an atomic or molecular scale. It's really a gene level scale. You get long range cooperative dynamics via DNA supercoiling, and they just did this directly. They were able to measure individual RNA polymerases moving along, and they were able to show directly this effect that you got interactions and speeding up of the ones behind the front based on this mechanism. And it's a sort of a nice single molecule experiment. And they pointed out that this doesn't have to only apply to the same gene. If you look at neighboring genes, just as just our calculation of the same thing. If you look at neighboring genes, nothing stops this mechanism from affecting neighboring genes. So let's say you have two genes that are right next to each other, and you're transcribing both of them. Right next to each other means within a few 5 kb or something along the genome. So if they're both being transcribed in the same direction, so RNA polymerase here is moving this way, RNA polymerase in this gene is moving this way, you get the same effect, that in between the two objects, you actually have a reduction of this effect. The second one can move more quickly than the first one, so transcribing the first one will positively upregulate the transcription of the second one. You get the exact opposite effect if they happen to be transcribed in opposite directions. If you're transcribing this way, if this gene, the RNA polymerase is going this way, and in this gene, the RNA polymerase is going that way, then the supercloning in between is adding, not subtracting. It's positive in both cases. And now you get an antagonistic effect. Now, if this gene is being transcribed, then that suppresses the gene next to it. And they, again, a number of people have shown this effect also in terms of tandem genes next to each other. And, we can predict this. So we described this in a number of papers. A number of other groups have worked on this. Now, let me come back to this noise issue. Now, the noise issue was already, again, argued qualitatively in this paper of 2014. It's called Mechanism of Transcriptional Bursting in Bacteria. And this was a case where they really just looked at circular. This was just an experiment that was done in an in vitro experiment. They took a plasmid, they attached it on the bottom, they added, topoisomerase, and they measured things as a function of how much topoisomerase they had, and they could show that actually, if you didn't have enough of this enzyme, and you went through this experiment, you would see increased bursting. And they understood that, without having an equation, they understood, and I'm not going to talk about the details. We can fit all their data using our model, but the answer is that if you have limited supply of one of these topoisomerases, one called gyrase, doesn't matter, then in fact you can create more bursting, and they show the increase of bursting as a function both of how much the promoter is transcribed and also how much topoisomerase they added into their experimental system. So the idea that noise somehow is related to this was already present, and our paper was actually meant to explain this data. So it was sort of, we got a freebie. We explained not just this data, but also some other data that came along in the meantime that wasn't possible. The technology to measure individual RNA polymerases moving along the DNA is not totally trivial. And so, okay. Now, I promised that I would talk about one, whoa, I always do that. Yeah. Yes. Your burst size increases, yes. Yeah, right. Burst, yeah, you can, yeah, so in the model, you can calculate whatever you want. Yes, yes, absolutely. No, in other words, it gives you super Poissonian noise in the technical term. So, yeah. Yes, but you need to know where the two states are coming from. But I thought that's usually ascribed to transcription factors binding and unbinding from. No, that's not enough. You need transcription, you need something which activates and deactivates transcription. It's not binding, is not going to give you that. I don't think so, anyway. Yeah, maybe you can. Okay, it depends on the system, but yeah, it depends how you treat binding. If you treat binding and unbinding as an equilibrium process, then it definitely doesn't give you that. If you treat it in a way, if you treat it fully as a stochastic process, it could give you something closer to that. But in fact, in that experiment, it's clear that that's not good enough to explain the data. You actually need something, and they show directly it's related to these topoisomerases. Yeah. Yeah. In this experiment, there was sort of only one gene there, so they didn't. So the answer is not in this case, but in the next case, I'll show you. In principle, yes. So I want to show you just a more, let's call it, hypothetical application of these ideas to a different experimental system. But yeah.
Just to clarify this point, so that picture of the gene state 1, gene state 2,
Yes. Yes.
if we solve that fully using...
Oh, you definitely get super Poissonian noise. Absolutely.
But that's not directly related to how many RNA polymerases are there.
No, but you need to know what determines the transition between gene state 1 and gene state 2. Right? I mean, in other words, you can solve that model assuming there's something that is gene state 1. And so, you know, I give this as a homework assignment, basically.
And that's just the transcription factor driven,
Probably, maybe, but but it isn't in these experiments. Yeah. So, anyway, so I have to think about. I mean, if you do this at, you know, using what people usually, just forgive the technical discussion. If you do this using what people usually assume and about transcription factor binding and unbinding, you could get something which is super Poissonian, but it doesn't have the same dependence on promoter rates and on transcription, on top-wise summarize. So, okay. So this is, I mean, it's not the only possible mechanism, but it seems to be relevant in some cases. Okay, I'll skip this, but you know, you have to do a little bit more work when you do this for eukaryotes, but basically it's similar things you can say. So I want to talk about one, okay, it's the same thing for eukaryotes. So I want to talk about this paper because, you know, there's an American expression, which is don't judge a book by its cover. I don't know how many of you have heard that expression. So here is a paper where I would say is a perfect instance of don't judge a paper by its title. Because I read the title of this paper and it sounded really, really interesting. You know, I will read it, you know, this is a paper in Science by a group that I know by a person I know quite well. A DNA repair pathway can regulate transcriptional noise to promote cell-fate transitions. Okay, that sounds really interesting. We've heard a lot about cell-fate transitions and how important they are. And cell-fate transitions, in many cases, depends on transcriptional noise. Okay, so far so good. So this paper is about what things can regulate transcriptional noise. And the idea is that transcriptional noise may not be just, you know, some mistake being made by the cell that it can't do something. It may be something which can be actively controlled by the cell in cases where it wants to be able to use noise as some useful thing. For example, if you want to make a cell-fate transition and you have a system that's sitting in some relatively stable state of your network, then one way to make that transition is to turn up the noise and have the cell be able to leave that state and then maybe find some other state, more differentiated state, for example, in a differentiation pathway. So this paper seemed to say that DNA pathways, that this was a mechanism that cells could use to regulate noise. Now, maybe it's true, but the statement that has something to do with DNA repair seemed really interesting, and that, I think, is the completely misleading part of the title. So let me tell you what they did. They took some system, and what they did is they added some chemical to the system that the DNA repair mechanism thought meant that there were, you know, base pairs that were being incorrectly incorporated into DNA as it was replicated. So DNA repair, just for there's a whole subject matter of DNA repair, which is quite interesting, quite important for the study of mutations in cancer, because one of the things which I don't think we heard about yesterday, but you know, when you say that cancers mutate and you assume it's at a constant rate, well, one of the things that cancers do is they actively dysregulate a lot of the repair pathways that are there specifically to prevent, you know, small breaks in your DNA from leading to mutations. And one of the reasons cancers can have a large number of mutations is because sometimes they just turn off these repair pathways. So DNA repair pathways is a very important subject. One of the types of repair pathways that exist is something called base excision repair. And it's a very technical field. Base excision repair is, in some sense, the simplest DNA repair you can make. You're going along, copying your DNA as your cell is dividing, and you accidentally put in the wrong base. The wrong base has some, you know, wrong chemistry. It doesn't really fit. So a enzymatic system can detect that. can bind to the DNA and can chop off the wrong base and basically go back and put the right base in. So it's called base excision. It's removing the previous base, and then it's going back, taking a step back and saying, whoops, made a mistake. I'm going to try again, and then just try to incorporate the correct base in the DNA replication process. Okay, now, one of the proteins that's involved in base excision repair is this protein APEX1, which is some large complex of things that comes and attached to the DNA. And one of the things that one of the components of APEX1 and one of these large protein complexes is an enzyme that has the ability to chop off the last base that you just added. And then, okay, so that's how it sort of works. I'm not giving the full description. I'm not even sure I understand the full description. And so he, added some, perturbation to the system that had the effect of convincing the DNA. So it's related to this particular protein. They added this molecule came in, attached here, thinking that it needed to repair the DNA, and it led to increased burstiness in the gene expression. Now, technically, and I think we said this before, technically, if you look at how people measure whether a expression pattern is bursty or not, is something called the Fano factor. The Fano factor is, by definition, or the coefficient of variation sometimes. It's sort of the ratio of the variance to the mean. And the reason why this is a useful measure is because for a Poisson distribution, if you ever took a course in probability, one of the first things they ask you to show, perhaps, is compute the mean of a Poisson distribution, compute the variance of the Poisson distribution, what's the ratio? And what you discover is the Poisson distribution has a variance which is equal to its mean. Okay, very nice. So that gives you a baseline for talking about whether transcription is noisy or not. So the definition of noisy or bursty or whatever you want to call it is that this factor, when you go and look at the rate of arrivals of messenger RNA, is greater or lesser than the baseline Poisson distribution. So when they say in this paper that adding this part of the DNA repair pathway is somehow causing increased burstiness, then that means that they measure this object and it goes up. So here is an example of their data. This is their data without this treatment. Here is their data with this treatment. You clearly see the increase in the range of fluctuations in the amount of messenger RNA in the system for some particular set of genes. They measure this actually for many, many genes. Now, the problem with when I read this paper, the problem with this title is one of the first things they did is they showed that the enzymatic activity of this complex, this APEX1 complex, that attaches to the DNA and is revolved in the actual repair process. If you get rid of the enzymatic activity by looking for, mutating it away or finding some way to get rid of it, it doesn't change their answer. So this has nothing to do with the actual ability of this enzyme to actually repair DNA. So what does it have to do with, right? If it doesn't have anything to do with DNA repair, first of all, that's why I complain about the title, but what does it have to do with? So our hypothesis was that what it has to do with is the fact that you've attached a very bulky complex to the DNA in many, many places. What that does is it pins the DNA at those spots. That is an example of one of these very large things that is so bulky that really prevents the DNA from rotating. So you've shrunk the regions over which the DNA can twist. Okay, if you shrink the regions over which DNA can twist, if you make the regions bigger, if I go back to my simple gradient picture over here, if these things are closer together, I get a steeper gradient for the same amount of angle change. And therefore, I increase the supercoiling. Therefore, I increase the burstiness. So we can take our model, we did take our model, and we could just run a simulation assuming that the only effect of this APEX1, well, it binds and unbinds at some rate, which you treat as a stochastic process. And the only effect of it was that it formed barriers that determined the lengths over which you could get this particular effect. And as I said, the barriers are what you need. You need to know where these barriers are in order to know how to relate the angle in which it turns to the actual amount of energy producing twist that exists in the system. And when you do that, whoops. When you do that, here is a picture from our model. Here is their data from, this was a yeast experiment on which they did this. Here's the data from our model. You can see, we took a whole bunch of different, this is their data, this is our model. When you take a whole bunch of different genes, you can plot, the mean of the gene expression. It doesn't change by very much. And then you can plot the fluctuation, the log of the Fano factor. And what you can see is that most of the genes have increased their fluctuations compared to what happens if you don't give this perturbation. So they succeeded in making a perturbation to this yeast cell. Yeast cells are fully eukaryotic in terms of their DNA structure. They have nucleosomes. They have all this higher order wrapping and whatever. And they showed that by perturbing it in this way, you could essentially have a large increase for almost all the genes that they investigated, many, and not just one or two genes. They looked sort of pan-genomic wide, and almost all the genes increased their noise. So it's a generic noise increasing thing you can do. But from our point of view, it's a generic noise increasing thing because this protein complex binds in many, places along the genome. It affects many, genes. And so you get a very pan-genomic effect by something which has nothing to do with DNA repair, but nonetheless is an interesting mechanism for cells to control the noise of their transcription. So what was the lesson that I wanted to, yeah, go ahead. Yes. And DNA, yeah. Yes. No, so here what this thing does is, so the way the competition works is it tries to turn the DNA. So the RNA polymerase, because it's getting bigger and bigger, so it's hard for it to rotate, it's basically turning the DNA in front of it. Now, if the DNA is not attached to anything, if I take my rope here and I just, and I just turn it like this, the whole thing just turns, nothing happens, right? It's, it's allowed to turn, nothing's the problem. The problem is if I hold it like this and make these points not allowed to turn, and then I turn it, then I have to have all the turn between this point and this point, and then I get this effect. So I need to have some type of barrier that prevents the thing turning somewhere. Then when I make it turn in the middle, I get this gradient of turning, which is equivalent to stretching. It's the rotational equivalent of stretching. So in this model, in our model of their experiments, what happens is the distance of those barriers moves they move closer together, and that means for the same amount of turning, it increases the force. So all the effects that I mentioned become increased. And in particular, the effect that they saw that we get from our model is the burstiness of the gene expression. And it's not a specific gene. It's not like this gene has been upregulated to be noisy. It's a mechanism for increasing the noise more or less across the entire genome. Now, they argue that that's important. They argue that if you want to make cell-fate transitions, you sometimes want to just increase the noise. Yes, so so there's no details about it. It just, yeah, so in other words, this is our hypothesis, and we can fit their data with this hypothesis. There's no direct measurement that, of at the single molecule level in a, let's say, in vitro system that this idea is right. That would be nice if they did that, but that's not what, they're not a single molecule lab, so they don't do that. But it's a simple explanation for what they saw, which one can argue whether it's important or not, but it does explain everything they saw. And so, you have another question? Okay. Yes. Yeah. Yeah. In other words, so maybe I'll draw a picture this way. So let's compare this picture to this picture. So let's say I have a typical distance between my preventing binding spots, my topological barriers. That's typically 10 kb or something. Then I come along and twist this, and this gives me some level of gradient in phi. Gradient in phi is called torsional strain. It's equivalent to really stretching something where you get a strain. And therefore, I get some amount of effect that, to move in here, I look up what the torque is. It sort of prevents the RNA moving into this region very effectively. If I then come along and say I'm doing exactly the same thing, I haven't changed the promoter, haven't changed the topoisomerase, all I've done is that I've taken my DNA, and now over here and over here, I've attached some big thing. So all of a sudden, the same, everything else being the same, gives me a bigger gradient than I had before, because I'm still forced to be 0 at this point. I can't not forced to be 0 at that point. So by shrinking the typical distance between these topological barriers to spreading of this twisting, I automatically enhance all the effects I talked about. I've slowed down on average, but also yes. No, so what they measured was the Fano factor. So in other words, they don't measure the absolute noise level. They measure the ratio. In other words, if I slow down the transcription, the mean goes down. So maybe the burst goes down too, but the ratio goes up. So you have to work, yeah, what? Yeah, so I have to see what, but yeah. Yeah, they did not measure the burst size. They just gave the overall statistics for the noise. Yes. Sure. Yes. No. So they would like to think the answer is yes, but they didn't show it. In other words, they would like. Well, so this lab, I think, has written a number of papers over the years, arguing, and this is a general question that I think the community has still talked about for many years: is noise always bad, or is noise sometimes good? Okay, so there's the noise is always bad philosophy, which says, why would you want to have random fluctuations? You want to have everything act like a perfectly engineered system. If you want to do something, you engineer it to do it. Why do you want to have noise? The other alternative is to say, you want some systems to somehow be able to explore different possibilities, see what's going on around you, and under some circumstances, having noise allows for a more efficient exploration, and the system can actually, in the end, do better. If you don't predispose what it's going to do, you let it figure out on its own what's going to do. That's a very, human-centric view of what cells are doing in this context. So this group, for a number of years, has tried to push the idea that having noise in systems like developmental processes that select cell fates, it can actually be a positive benefit. So if it's a positive benefit, then you would want it to be regulated, because obviously there are other cases where noise would not be good. If you're sitting in some, let's say, epithelial state in the middle of your lung, and all of a sudden it starts being very noisy and you have a chance to become mesenchymal because of the noise, that wouldn't be very good. So you don't want noise to always be large. You want it to be regulated. So they've looked for mechanisms that can that if they assume that noise is good for you under some circumstances, the next step you'd want to find is a signaling network or a process that can turn it on and off when you need it. And they're thinking about development usually in their experiments, even though they're using yeast. So what's development in yeast? But nonetheless, they're thinking about it in terms of their conceptual framework. They're thinking about it in developmental systems. So that's what they're proposing. We have no professional opinion about whether that's a good idea or a bad idea. We're just saying that the actual mechanism whereby you have this increased noise, at least one can use this model to try to understand that and see if this is the right way to think about it. Yes. Yes. Well, the topoisomerase is just, you can think of it as just a totally decoupled, in our model at least, it's a totally decoupled mechanism by which supercoiling relaxes over time. So we don't put in some effect that says the topoisomerase knows that there's a traffic jam here and it goes specifically to there, because that we don't put, maybe it's true, but we don't know of anything that does that. We just say that the topoisomerase is a general relaxation mechanism for supercoiled DNA, and this is, and so it's it does bind supercoiled DNA because that's its job. So if you create regions of strongly supercoiled DNA, the topoisomerase will bind there and will have an effect, but it's not specifically looking for traffic jams. It doesn't know that it should bind here because there's 7 RNAs waiting in line to proceed. It just knows that this is a region that has strong supercoiling, and so it binds to that and changes the supercoiling density. So at least, okay. So it's less intelligent than, maybe it's, at least in our model, it's certainly less intelligent than it might be. Okay, so let me just give you a preview of, okay. So this was the first part of, and I think there's a little respite from hearing about data and about, genes. Well, we did hear about genes, but we didn't hear about networks of genes. We didn't hear about, I don't know, bioinformatics. But, I think there's I wanted to go through this. First of all, it's sort of a nice story, I think, because, you can do something and you can try to understand something from a little bit more of a, let's call it biophysical perspective than many other things that are out that people are looking at. And it does remind you that, the DNA is not just a, I don't know, tape in a Turing machine or something, or something like that. It really is a physical object, and sometimes the physicality of the object plays a role in what you're thinking about. In particular, I said many of our models of cell-phase transitions, such as the EMT models that you heard about yesterday, such as some of the other models, are, in the end of the day, assuming some level of noise. You typically need noise to accomplish transitions in some of these models. And so the noise has to come from somewhere. It can come from just binding and unbinding noise, but at least in some cases there can be, let's call it, other phenomena that can modulate the noise in various ways. You got a question? No, okay. Okay, so so this is just the first part, and I'm of course gonna quit, but just talk about the next part. So we already heard, a million talks about epithelial plasticity. This is the poster child. This is, in fact, the picture that in some sense let us start working on this subject some number of years ago, where you have this picture due to Bob Weinberg's lab that you have this primary tumor and cells undergo this transition and leave and go through the circulatory system and wind up somewhere else. And and I'll talk about this a little bit more tomorrow, but I want to just mention what I'm going to talk about. So I'll go through a little bit more detail about how we do this and about experiments, but I want to get to the point here. But it became clear to us that we were missing part of the story, and that's the story I'm going to tell tomorrow morning. So tomorrow morning I want to tell about the missing part of the EMT story that you haven't heard about yet. Now, what is the missing part? So I want to describe one experiment, and then as a warm-up for tomorrow. Here is an experiment. So this is a paper called, whoa, I always do, I always have this problem with my cell phone. I always press the wrong button. Okay, back to this paper. Maintenance of epithelial traits and resistance to mesenchymal reprogramming promote proliferation in metastatic breast cancer. Okay, a nice title. Let me translate what's going on here. So this is a particular cell line. HMLE is human mammary cells. Okay, so normal breast cells in humans, and they took those cells, they put in an inducible expression system, which was just engineered, and the inducible expression system allows them to express TWIST. TWIST is an EMT transcription factor. It's one of many, but it is something which, if it's upregulated, and its products will produce EMT in the cell. So they have a controllable system where they can induce EMT. So what do? They induce EMT. And what do they discover when they induce EMT? Well, here is their system, twist ER, whatever. And what they discover is that they start out with cells. Here they're all in this epithelial box. That's this box over here. And with 90, whatever it says here, 96.3% of the cells are positive for this EpCAM, which is an epithelial marker, and then they turn on EMT, they induce it. This is by tamoxifen, and almost all the cells move to the left, to the right in the figure over there, to the left when I look at it, which is that they reduce their epithelial marker, and those cells, as well as you can ever say with just one marker. I mean, I think we heard already in many cases that one marker is not a great way to define anything in a biological system, but nonetheless, they use one marker. And from their way of looking at it, the vast amount of the population moved into the other box, into the clearly mesenchymal box. However, there were 4% of the population that did not move into the box. So there was resistance to EMT by about 4% of the population. Now, okay, why do you care? Now it turns, so they call these things, ESSCs, and these were MSSCs, but it doesn't matter. But the actual interesting thing here was when you read the paper carefully, this is one marker, but they did actually measure some other features of the cells, like cell shape, for example, that we heard a lot about earlier. And what you really see is that those cells that are in this box, it's not that they didn't undergo EMT at all. It's actually they underwent some form of partial EMT. In other words, what they measured here is the loss of epithelial traits. And partial EMT, as we've come to understand it, is usually manifested itself with when you retain your epithelial states, your epithelial traits, and you pick up mesenchymal traits. Okay? So that's exactly what these cells did. Here you see only the loss, the lack of loss, the retention of the epithelial traits, but actually those cells now also have some mesenchymal states. This was before the era where everybody in the cancer biology world believed in partial EMT states, so they just called these states mesenchymal. Fine, no problem. But then what they showed was if you took these cells after the sorting experiment and injected them into a mouse and generated organoids, and then actually only these cells, the recalcitrant ones, or the ones that we would now call partial EMT cells, are the ones that were able to generate new growths in the mouse. Now, from our point of view, this is fine, because this is exactly what all those other papers that I cited yesterday have also shown. It's done, in a slightly different way, but it's consistent with the general picture that we expect states that have only undergone partial EMT to be more effective at generating new tumors, and in this case, new growths within a mouse. So it's all fine. There's nothing that contradicts anything that we've been claiming about EMT transitions for a few years by now. But the real question is, why did this happen? What was the difference between those cells? Right? You started, yeah? Are different from the original state? Yes. No, they're at the level of resolution of this experiment, they just look like hybrid states. So the hybrid states are the state, and we tend to call the process partial EMT, but it's just terminology. Everybody has their own terminology. So yes, no, they basically look like hybrid states in our models, that they have not lost, they've maybe reduced a little bit things like ecadherin or EpCAM, but now they have some vimentin, they have some other mesenchymal markers. So they look like, some real world version of the simple model hybrid states. There's always the real world version versus the model version, but yeah. So the question then, which you obviously listen, you read this paper, and the question is not, that these cells are able to grow and these are not, that we think we understand. The real question is, what was the difference? They took what was supposed to be an isogenic cell line, they did the same experiment to all the cells, and yet 5% of them did something different than the other 95% of the cells. And they didn't really answer the question, but they tried to. And the one thing they said was, so here is a quote from their paper, the ability to retain these epithelial traits is tied to a global epigenetic program that limits the actions of the EMT transcription factor ZEB1. Let me translate that, and then I'll quit, and we'll talk about this in great detail tomorrow. So underlying everything that's been told to you about the networks that you've heard about is the fact that the structure of the DNA, in particular now we're talking about the post-transcription modifications of either the DNA by adding methyl groups or the histone proteins by adding either methyl or acetyl groups or other things. All those modifications are involved in modulating how easy is it for a typical transcription factor to come in, attach to the DNA, and begin to actually make the RNA that we care about. So there's a regulatory layer that's above or below, whichever way you want to think of it, different than the regulatory layer of the genes, the transcription factors, and the other factors regulating translation that we have heard about, and you'll get a brief review of it tomorrow from me. But we have not yet talked at all in this whole conference about this extra regulatory layer. Now, the only evidence they had for this regulatory layer was some preliminary measurements using ATAC-seq. For those of you who don't know what ATAC-seq is, it's a measurement of DNA accessibility. It's not specific to any transcription factor or whatever. It tries to just determine if the DNA in some region looks like it's open and accessible versus closed and inaccessible. And what they saw when they tried to measure this is they saw that some of the traditional EMT transcription factors, for example, ZEB1, which is one of the ones that's most studied in most common transcription factor for inducing EMT, that the DNA structure around ZEB1 was something that looked closed off. It was inaccessible. And therefore, that, ZEB1 could not, I think I said this quite the wrong way, that the places in the genome where ZEB1 is supposed to bind to get rid of the epithelial cells, ZEB1 suppresses the epithelial cells, were inaccessible. So ZEB1 could not do its job. Even though it became higher in concentration, it could not turn off the epithelial traits. And this is great, but this is all they said. And so what were we supposed to do with this? I mean, it's hard to make a model based on one sentence in a paper. So we didn't. But the interesting thing is that biology experiment keeps catching up to all our ideas, which is really great. And there was a new experiment done about 3 years after this one that did something very similar to this, but actually made a lot more technical progress in measuring things. And that led us to what I will talk about tomorrow, which is, I would call, a very preliminary attempt at understanding how to include this other aspect of DNA, this histone mark aspect of DNA and its accessibility based on those histone marks into the types of network models that we've been discussing for EMT and other cell-fate transitions. So with that, I'll quit. It's about 3:30, which is, I guess, around the right time. And I hope you enjoyed this, what is the world diversion into sort of more biophysics, because I think it's good for you. And tomorrow we'll return back to the world of cancer and back to the world of regulatory systems, but with a perspective that there's something about the physical nature of the way DNA is packaged in terms of these nucleosomes that becomes increasingly important when one thinks about networks and states and transitions and even metastasis. Thanks. Happy to answer more questions. Yes. Sure. For here or for the other case? So, so some of the parameters we know, so a lot of the parameters were known from single molecule experiments. For example, the curve of how the velocity depends on torque. So you can measure this in a lab. You can actually take a piece of DNA, attach it between two beads, twist the beads, so put a twist in the DNA, put one RNA polymerase on it, and watch it move. You can actually do that in the lab. It was done in the lab by Michel Wang at Cornell. And you can actually measure how much the velocity will depend on this twisting. So some of the parameters are gotten directly from these direct biophysical measurements. Some of them are not known, or at least not known very exactly, and those we had data from this Cell paper that I mentioned. There were a number of other experiments that have come along since then. So in some cases, we just try to fit some of the unknown pieces to some experimental data. So, but a lot of the parameters, a lot of the basic biophysical parameters are known, because people have done single molecule experiments on this, and and so a lot of it is known, because it's not really the full complexity of biology. It's really a focused question: how fast can RNA polymerase move on DNA? So you can do that in a lab and expect, with some caveats, you can get an answer which then can be interpreted as to what the answer would be in an actual cell. So there are these single molecule experiments which then are ongoing, and and to measure these parameters in detail.
Any other questions? Okay. If not, we can continue outside. So the plan today, since not enough of you volunteered to give talks, it would have been great, but so therefore we've canceled today's presentation session. So we move for coffee, and then it's free time, but feel free to go to the posters and hang out and discuss and chat with each other. Okay.